So let's talk about oogenesis. Oogenesis is the formation of the egg. Uh, in humans, the formation of the egg is quite peculiar compared to other vertebrates and compared to other mammals. So first of all, the cells that produce the eggs in females of our species, the PGCs, are already present in the developing embryo. In fact, the number of PGCs increased to almost 7 million before birth. But almost near birth, many of these cells undergo apoptosis, which is programmed cell death. And after birth, we have more or less 500,000, which are all the eggs, all the cells that will produce the eggs for the rest of the life of the female of our species. Now, egg production really initiated puberty thanks to pulses of retinoic acid. So again, retinoic acid is the signal that initiate the egg production, or if you want, meiosis in this case. Now, meiosis of these PGCs start the puberty, start after pulses of retinoic acid, and of course the first thing that happens during meiosis is duplication of DNA, so synthesis of DNA. However, almost immediately after synthesis of DNA, when the sister chromatin are exchanging DNA, so if you want, during the crossing over stage, the meiosis is blocked. So all the eggs that have started their meiosis are blocked in what is called the dictyate resting stage. And these eggs that are blocked in meiosis can stay in this stage for the rest of the life of the person. So it can stay there for 1 month, 1 year, 10 years, 20 years, 50 years, for the rest of the life of the person, or until it goes to the person goes to menopause. In humans, of course. However, every month, pulses of luteinized hormones induce some of these eggs that are blocking dictyate resting stage, to progress in their meiosis. So, luteinized hormones induce further development, further progress in the meiosis, and the meiosis progress to metaphase 1, to anaphase 1, and telophase 1. And during ovulation, the eggs that are released from the ovarium in mammals, in humans, are actually in humans, not mammals, are actually just beginning meiosis too. So they are secondary oocyte. So they are oocyte that did not finish meiosis. Indeed, they are just starting meiosis too. And the other thing that are interesting to note here is that only the eggs that are fertilized will finish meiosis. So if the eggs are not fertilized, they don't even finish meiosis, and they are removed, released, abandoned in our species even before they finish meiosis. In fact, they don't progress from a secondary oocyte unless they are fertilized. So here we are. One further important peculiarity is that differentiate the meiosis of eggs compared to the meiosis of spermatozoa is that the cells that are produced during meiosis 1 and 2 are not all of the same shape. In fact, because of the spindle is not in the middle of the cell, but the spindle is pushed toward one side of the cell, as you can see here. So when the cells divide after, for example, the first meiosis, meiosis 1, we have the formation of two daughter cells of different size. One daughter cell that is very big, that contains most of the cytoplasm of the original cell, and one small cell that contains very little cytoplasm, which is called a polar body. Now, in our species, fertilization occurs at this stage. So the secondary oocyte are released from are released from the ovarium. Fertilization occurs when the egg are initiating second metaphase. So at the end of meiosis, in our species, in other species as well, what we have is the formation of a big egg, which in our case will already be fertilized, and three polar body. The polar body are very small cells, and they are not really used for anything. So what can we find inside a mammalian oocyte? So this is not only our oocyte, it's in general mammalian oocyte. So if you look externally, the egg is covered by a layer, a very thick layer of follicular cell, which is called the cumulus. The cumulus. So if you want, is the egg or the ovulum, and it is surrounded by a layer of cumulus. Nearer to the egg, or approaching the egg, the follicular cells become more thicker. And here, this thicker layer of follicular cell is called the corona radiata. Below the corona radiata and near the membrane of the egg, we have a matrix of proteins that is called the zona pellucida. The proteins that form this matrix are called the zona pellucida proteins. And below the zona pellucida, we have the membrane of the egg. Inside the membrane of the egg, we find the nucleus, if you want, of the egg itself. So what do we find inside the egg? Well, we find the mitochondria. So in our species, in many other mammals too, the mitochondria come with the egg. So the paternal mitochondria usually do not take part to the formation of the zygote. However, time to time, some of the mitochondria some of the mitochondria from the spermatozoa are able to enter the egg as well. But this doesn't happen very often. Other things that we find inside the oocyte are nutritive proteins that are called yolk proteins. These yolk proteins are produced in the liver of the mother and are deposited in the egg. Now, in most mammalian species, there are not many yolk proteins because, of course, the egg is sustained. The fertilized egg is sustained by a forming placenta. But in other vertebrates like frogs or reptiles, for instance, or birds, their embryo is sustained by big yolk that are deposited indeed in the egg during the egg formation. Inside the egg, we also find everything that is needed for transcription and translation of genes and proteins. So we find messenger mRNA that are deposited by the mother, ribosomes, ribosomal RNA, ribosomal proteins, transfer RNA as well. We find, obviously, a lot of messenger RNA that the mother deposits so that the embryo can progress through the first correct path of cell divisions. And we have all the other morphogenic factors that control the expression of the genome of the embryo itself. Okay, so here we have a sketch of the ovaries. Here you see the egg that is released from the ovary, and the egg that is released from the ovaries is surrounded by the cumulus. Now, the egg enters the ampulla. The ampulla is the higher part of the female tubules that connect the ovary to the uterus. In the ampulla, high in the ampulla, we have fertilization, and then the fertilized egg is translocated, a process of translocation, from the ampulla down the isthmus and then reaching the uterus. Fertilization, you can see here, occurs very high in the ampulla. In our common imagination, we think that the sperms that are released from the male swim toward the eggs. Which is only partially true. In fact, for the most of the journey, the sperms do not really swim. They are transported by the maternal female reproductive organ muscles. So sperm motility, is important in two main points. Initially, in the cervical mucus, so the sperm swim toward the internal part of the female reproductive organ, and then at the end, very near the egg. For the rest, it doesn't swim at all. It is actually transported by the uterine muscle contraction. And in fact, sperms can be found in the oviduct here, in the isthmus, 30 minutes after deposition in the cervix. So it is not possible for the sperm to swim that far so fast, and in fact it is because it is transported by the uterine muscle contractions. Even now that the sperm is incapacitated, so they don't have the capacity to fertilize the egg, they demonstrate what is called sperm rheotaxis. So sperms are able to swim against the flow of liquid. Because the flow of liquid is from the internal part of the female organ to the external part of the female organ, so the sperm is able to swim against that. So they're swimming toward the internal part of the female organs. As I mentioned already a couple of times, these sperms do not have the capacity to fertilize the egg. So they are not capacitated. And in fact, even sperms that arrive to the ampulla too quickly, so therefore are not capacitated, are wasted in a sense, so they are unable to fertilize the egg. However, on the opposite side, the capacitated sperms survive for a very little period of time. So almost very quickly after that they acquire the capacity to fertilize the eggs, the sperms are dying. So in order to increase the fertilization window, the oviduct of the mother, the isthmus in particular of the mother, capture the sperms, allow them to capacitate, and then release them in packets. So instead of release them all, they release them just a packet of capacitated sperms near the egg. And then because the oviduct will release multiple packets, this increases the fertilization window. Okay, now we mentioned capacitation a few times, but what is it? So during the capacitation, the sperm acquire the capacity to fertilize the egg. This capacitation occurs or happens during the journey of the sperm through the ampulla, toward the ampulla. Capacitation is important both because it permits the acrosomal reaction and also it permits the hyperactivation of the sperms. So what's happened is that the cholesterol that is present in the membrane, in the surface membrane of the sperm, is removed by the albumin that is present in the female oviduct. So the female oviduct express albumin, they attach the cholesterol, remove the cholesterol from the sperm membrane. And when this happens, this removal of the cholesterol works as a signal that change the membrane of the sperm, and also a signal for changes in protein inside the cytoplasm of the sperm. First of all, when we remove the cholesterol, some proteins that are receptor for the zona pellucida are exposed. So while the cholesterol was there, the receptor were not showing on the membrane, they were hiding, if you want. But when we remove the cholesterol, this receptors are now exposed and they can bind the zona pellucida. Without removing the cholesterol, this receptor cannot bind the zona pellucida and therefore there cannot be fertilization. The removal of the cholesterol works also as a signal internal of the cytoplasm, and a lot of protein in the cytoplasm are modified, and some of these modifications are important because this allowed some of the proteins that are in the cytoplasm to move and be exposed on the surface of the membrane. These proteins usually are important either for the binding of the zona pellucida or the binding of the membrane of the egg. So, exposing this cholesterol, exposing this protein after removing the cholesterol is essential for the acrosomal reaction, as we will see in a minute. This removal of the cholesterol make a dramatic change in the membrane of the sperm. This dramatic change is translated to a signal that will modify different proteins in the cytoplasm of the sperms, and some of the proteins that are modified that can now move from the cytoplasm to the membrane of the sperm. One of these proteins is izumo. Izumo is essential, I will see in a second, for sperm-egg fusion. Without izumo moving to the surface, we will not be able to fertilize the egg. So, incapacitated egg is connected, if you want, is bind to the isthmus, and it stay there until it become capacitated. And only capacitated sperm are released in packets toward the egg. So we say that this increases the fertilization window, but also this is a block of polyspermy. What is block of polyspermy? So what is polyspermy? Polyspermy is the occurrence of that we have when multiple sperms fertilize the same egg at the same time. So if multiple sperm fertilize the same egg at the same time, we have an excess number of chromosomes. So we have polyploidy, and our embryo cannot survive. So mammalian embryo cannot survive if you have too much polyploidy. Therefore, to avoid multiple fertilization by different sperms at the same time to the same egg, what's happened is that eggs are released in small packets. Sperms are released in small packets. So imagine that we have a lot of sperms around the egg. If you have too many sperms around the egg, this will increase the chances of to have polyspermy. But if you release just small amount of sperms near the egg, then the chances to have polyspermies are reduced. So this block of polyspermies work in this way. Instead of having all the sperm released at the same time, we have just little group of sperms released, and if the fertilization doesn't occur, another group can be released. In this way, we not only increase the fertilization window, but also we reduce the chances of polyspermy. So, capacitated sperm become also hyperactivated. And this is because during the process of capacitation, some ion channels are open in the membrane of the sperm tail, and this allow them to move strongly. So the sperm tail become started to move more strongly with bigger angle, so the sperm can swim faster and in a rotational way. So instead of swimming just on a plane, it started to move on its axis. So it started to rotate this way. So in this way, the swimming become more efficient and they swim faster. Only capacitated and hyperactivated sperm can now recognize and sense the last few clues to reach the egg. One of these clues is temperature. So this is sperm thermotaxis. So only capacitated and hyperactivated sperm become sensitive to temperature. There is a difference in temperature between the isthmus and the ampulla, and the ampulla is 2 degrees higher in temperature than isthmus. So hyperactivated sperms and capacitated sperm recognize this difference and they swim toward the warmer pit where the egg is supposed to be. Finally, the cumulus of the egg is releasing chemicals, molecules, that attract the sperms, but only hyperactivated and capacitated sperms can recognize these molecules. And this again is because some of the receptors for these molecules are now available on the surface of the sperm's membrane, and they were not there if the cholesterol is present. So if the cholesterol are blocking all these sensors, but when we remove the cholesterol, all these sensors are available and therefore can recognize the molecules that are produced by the cumulus. And this is a process called chemotaxis.